Thank you, everyone. We're going to start this session. We have an esteemed panel here with us today. Everybody come on and get seated. So this panel is regulatory pathway and applications in drug development for rare disease and case studies. So the purpose for this panel I alluded to yesterday. There's been a lot of recent approvals that, they disseminate into the broader community. There's, misconceptions sometimes about the totality of evidence that get discussed in the broader community. So what better way to address this was to assemble a group of people that could speak to different elements of, recent approvals. Obviously, there are some individuals that can't speak directly to their product, but they can talk through the steps on this panel. But what we wanted to do is address those so that people would get an opportunity to see, and hear examples of the dialogues and conversations, but also the levels of evidence that are needed to be done. So I'm going to start this panel with continuing the conversation we had regarding Reata, and Shannon, if you want to interject anything additional after you hear comments from the agency. We have Emily here, who actually did the review and can provide some background experience, and we'll come back and we'll interview people. And after this, I just want to start that for continuation, and we'll go down the row.
Sure, thanks. So for those that don't know me, I'm Emily Freilich. I'm the director of the Division of Neurology 1 at Cedar. And this is a good continuation since we just heard a lot about omaveloxolone and Reata. And I think Dr. Baracchio also mentioned yesterday some of the considerations that went into determination of substantial evidence of effectiveness for that application. As you heard, we have to determine substantial evidence, and typically it can come from two studies, a single study, or a single study plus confirmatory evidence. And as we also heard, Reata, omaveloxolone did have a positive, adequate, and well-controlled study that won on its primary pre-specified endpoint. But it had some limitations as well. It was a small study. It did not have positive findings on the secondary endpoints. And so it wasn't persuasive enough to stand alone as a single study. And so we really were considering what could serve as confirmatory evidence to support what appeared to be a positive treatment effect in the single pivotal study. And they had presented also some mechanistic data that showed that the drug seemed to be doing what it was purported to do. So some biologic plausibility that the drug was having its effect on NRF2 activation. And the natural history study, which we heard a lot about in the last session, but the benefits of that study, or the positives that allowed us to really rely on it, were what we heard a lot about was the natural history database, right? This FACOMS, where we had individual patient-level data and from a large number of patients that had systematically collected the same mFARS that was the endpoint in the primary, in the open label extension for the pivotal study. And so the other thing that was also compelling was that a lot of the centers were the same, and so we felt confident that the data was collected in a similar fashion. I think there was overlap on a lot of the centers between the natural history study and the open label extension study. And so looking at that data and seeing that you had several years of progression in the natural history data and could compare it to the 3 years of open label extension data on the same endpoint on the mFARS, and we're able to do the propensity matching because they had collected demographic data for baseline characteristics to be able to match on the important prognostic variables. That was really what led us to be able to rely on the natural history study for confirmatory evidence. Now, there are always limitations to external comparisons, and this was not immune to that. It was not pre-specified. It was an exploratory analysis, but it was able to support the primary endpoint, and that was really how we were able to rely on it. It would not have been able to support efficacy of the drug in and of itself, as many open label external controls studies cannot do. Although, in rare disease, that is an option sometimes to do an externally controlled trial, and FDA does have an external controls guidance that gives you kind of the considerations and limitations of using external controls.
Do you want to comment any more about that and other applications that you've reviewed and use of real-world data? I mean, any examples that you saw that were really effective or ones that might be more challenging? And you can keep it more generic and not throw somebody under a bus, but we'd like to hear more.
Yeah, I think one thing to consider, and this is not necessarily how we're thinking about it today, but the agency has been using real-world data for many years to look at safety, right? Post-marketing, we look at real-world safety data all the time to update labels, figure out when there needs to be a warning. And so we're not strangers to real-world data. There's also been a recent example in the Division of Neurology 2, where they were able to use real-world data to update the dosing regimen because there was safety data about the safety of that dosing regimen for an epilepsy drug. So I think real-world data use is out there. When we get to efficacy, it's a little harder because of the limitations. But I think if you can pre-specify to the best of your ability what you're going to be looking at in the natural history study, systematically collect the data, in a similar fashion at a similar time points that you're going to do in your controlled study, and having that patient level data, those are some of the key things to consider. And you can't control for all the confounding and bias, but the most you can do to limit that bias, the more successful you may be in having an externally controlled study.
We'll just continue on with introductions of that. I just wanted to not lose that thought, and since we had the break, it would still remain in people's short-term memory. Steph, do you want to go ahead and
Yeah, I'd be happy to. Hi, everyone. It's a pleasure to be here. My name's Steph Fredette, and I work at Biogen. I oversee the clinical development aspects of our neuromuscular portfolio and led the development of the tofersen program for SOD1 ALS. And then my new favorite role that I just took on is a co-chair of the AMP-ALS initiative that you heard a lot about yesterday. So it's an honor to be here, and I look forward to the discussion.
Hi, I'm Dawn Phillips, Senior Director of Clinical Outcomes Research at Regenix Bio, and I'm excited to be here and to have this opportunity to share how we do endpoint modeling within some diseases I work in. I focus on rare. We have 4 rare disease programs, and I work across them.
Hi, I'm Allison Martin. I am a director on the regulatory science and policy team for North America at Sanofi. I span the different TA areas, but focus pretty heavily on patient-focused drug development, rare disease, and real-world evidence.
Hi, I'm Jessie Hanrahan. I'm the Chief Regulatory Officer at Solid Biosciences, which is an AAV gene therapy company dedicated to the development of neurologic and cardiac therapies for patients, including Duchenne muscular dystrophy and Friedreich's ataxia. Prior to my time at Solid, I worked at Bluebird Bio, where I oversaw the global regulatory strategy for their severe genetic disease programs, which included Zynteglo and Skysona, which were recently approved here in the United States. And prior to that, I worked at Genzyme across MS and oncology and rare disease.
Hello, my name is Larissa Laptila, and I'm the Associate Director in the Division of Clinical Evaluation and General Medicine in the Office of Tissues, in the former Office of Tissues and Advanced Therapies, but currently Office of Therapeutic Products in the Center for Biologics Evaluation and Research. And many of you probably know that our office oversees, although we changed the name, we oversee the same categories of products, and they include cellular therapies, gene therapies, tissue-engineered products, plasma-derived therapeutics, xenotransplantation products, and a number of other biologics. And most of our product development programs that we see are, in fact, for rare diseases. And I also look forward to the discussion.
Hi, my name is Mark Rudd. I work in Global Regulatory Strategy for Biogen. I oversee our neuromuscular disease franchise. So Steph Fredette and I work very, very closely. I'm her regulatory to her clin dev. We do get on very well. We just decided to sit a few
She told me to sit you over
space.
there.
We work together for
Just
about...
told you to separate you two.
The biggest privilege I've had over the past 18 months has been to lead the CARSODI new drug application and in the advisory committee meeting, which Steph moderated and presented on. So we have a lot of experience in rare diseases across AbbVie, Celgene, and GSK previously. And I said most recently, the one we'll probably talk about a little bit more about today is CARSODI, the accelerated approval in SOD1 ALS. And then we'll be friends again, right?
Okay, given that, Steph, I mean, there was a lot of excitement after the advisory committee and the subsequent approval of tofersen. Could you start telling the story and sort of building the level of evidence, and then we maybe can have FDA react to the readout, I guess, from the discussion.
Absolutely. I'm not usually organized enough to take notes, but Mark reminded me that brevity is not my strength when talking about tofersen, so I figured I would just write a few things down. But it's been an incredible journey, and one that started far before I was working on the program. It's in 1993, SOD1 was the first gene that was identified to be relevant in the pathophysiology of ALS, and it's an ultra-rare subset of the population it affects. I should say there are approximately 300 people living with symptomatic SOD1 ALS in the US right now. And 30 years later, in April, we had the first targeted therapy for the treatment of SOD1 ALS approved with tofersen. It received accelerated approval based on reductions in neurofilament light that were found to be reasonably likely to predict clinical benefit. And just very briefly, tofersen is an antisense oligonucleotide, and it's designed to bind and degrade SOD1 protein. It's trying to reduce the protein and the accumulation of that protein to preserve sort of the function of motor neurons. But I'll jump to the phase 3 study because I think there's so much we can learn. We knew that neurofilament was important. It's a marker of motor neuron health of the active neurodegenerative process, and so we were measuring it as a key secondary endpoint. But it was not our intent to pursue accelerated approval when we designed VALOR. We were hoping to demonstrate substantial evidence of effectiveness on a clinical outcome measure, the gold standard endpoint, ALSFRS-R, that I could talk about for hours if anyone wants to after this, in ALS trials. And so off we went, and we designed a 6-month trial with an enriched primary analysis population, which we thought was the right thing to do at the time. And we had, unfortunately, an open label extension that I'll talk more about. And we designed the study based on the very limited natural history data that we had available to us at that time. And come to find out, we vastly underestimated the size of the study that we needed, and that's been an important learning as well. Not only is there limited natural history data, but it's also not consolidated, not collected in a consistent manner, just again emphasizing the importance of everything that the Critical Path Institute and NIH and others are trying to do. So in August of 2021, I saw the primary analysis, and I don't think you can get a p-value worse than what I saw that day. I thought it was a joke. I actually did. I thought my statisticians, some of you may have heard me say this before, were playing a joke. There were many tears, and then the next day I saw the biomarker data. I saw the reductions in SOD protein, and I saw the neurofilament data. And at the same time, I got transcripts from these qualitative exit interviews that Dr. Campbell actually recommended we collect in the VALOR study when we were having conversations about the phase 3 design. We didn't include them as quickly as I wished we did. We only had a handful of these exit interviews, but I was reading them, and I remember distinctly having tears streaming down my face because I was lining up the biomarker data and the exit interviews. And these participants were asked to weigh in on how has the disease impacted their life and how has that changed since they started participating in the trial. And they were so eloquent and so specific. A couple months in, nothing changed. But 2-3 months later is when I started to feel different than when I started. And looking again at the time course of the biomarker effect, I called my statisticians and I said, my goodness, it's working, and the trial design is all wrong. And so we had a long journey of being humble and really just trying to learn from the data, learn from conversations with regulators and the clinical community. We, a couple months later, had data from our open label extension looking at integrated analyses, early start versus delayed start analyses, so highlighting the importance of maintaining the blind from your parent study when you're conducting open label extension studies so that you're preserving the integrity of data collected in that fashion. And Mark can share more about where this bundle of fun that I handed him on the regulatory side to try to navigate. But ultimately, to jump to the sort of punchline, it was the totality of evidence, the pathophysiology of SOD1 ALS being relatively well understood, the mechanism of tofersen being understood, and really importantly, our understanding of what neurofilament is, what it's telling us, and the massive amount of work that the community had done to progress our understanding of neurofilament in ALS and identifying it as a prognostic marker of disease progression and survival that led us to ultimately having a successful advisory committee meeting and accelerated approval. The benefit of tofersen will hopefully be confirmed through the ATLAS study, which is the first interventional study, as far as I know, in pre-symptomatic gene mutation carriers. And as my friends at End the Legacy always remind me and others, is it's not a different population, it's an earlier part of the disease spectrum that we're evaluating, and likely the part of the disease spectrum that we need to be intervening in to really have a more profound impact on the disease. And so we're hopeful there. And the last thing I'll say is that while hitting the primary endpoint would have made life much easier, I really don't think that we would have learned as much as a community, certainly I wouldn't have, if that had happened. I feel like I've heard someone say that we broke the system with tofersen and really forced conversations that are happening now about what is the proper trial design, the importance of extension studies, the importance of following the science, the importance of collecting objective measures and all that. And I'm happy to expand on any of those points.
Yeah, you know, one of the things I noticed in your conversation is the amount of discussions. You didn't highlight as much the NFL parts of that, and that seems to be the most buzz,
Yeah.
you know, people talk about. So for both of you, like, how did you prep for this, you know, when you're talking your regulatory strategy about inclusion of NFL? You know, what homework did you need to do early? You know, what types of preparatory things for your arguments did you think things through? I mean, any of that would be you know, that you can share would be greatly appreciated.
Yeah, I can start, Mark, with the sort of why did we include it as a secondary endpoint? I mean, I would say I spent a good amount of time working in the spinal muscular atrophy world prior to coming to ALS. And so I had a pretty profound appreciation for the importance of measuring neurofilament and what it could be telling us about the disease. But again, it's the work that the community had done to characterize it as a prognostic biomarker. So we were confident that it was important, and it was actually our second key secondary endpoint, which is a bit unusual in the context of a clinical trial. So it was part of our hierarchical testing and such. And so that part of it was pretty clear and didn't require a lot of conversation. We needed to be using the right assay and making sure that we were assessing in the right way. But then ultimately preparation for the advisory committee meeting, I can have Mark weigh in on sort of what some of the fun that went into that, and of course discussions with the agency.
No, I'd say the most important part for the team was that kind of evolution from when the phase 3 study really read out. So everybody in this room knows that drug development is not linear, and it's certainly not for rare diseases. But most importantly, post-study readout, we had to really work on how we were defining substantial evidence of effectiveness. How could we then best position the NDA for the agency to begin their review, as well as hopefully lead it to a successful outcome. So when we first came to the agency, the first discussion was around the feasibility of this data set in its totality of approach. Colin, as you mentioned in some of your introductory comments, when you had strong target engagement, you had substantial tofersen-driven reductions in the biomarker, you had clinical trends that were numerical in an ultra-rare disease setting. And I think we had a very important discussion with the agency early that stated, you know, in support of a traditional approval, one must show substantial evidence of effectiveness on a clinical endpoint or a verified surrogate endpoint, which we did not have, as Steph discussed. And then really, as we were able to take the agency's feedback and really go back and look at our evolving data set, evolving because we also got to look at the open label extension data that really enabled us, with some statistical limitations, to look at the patients at 52 weeks. And I think we saw some really interesting data in there. You saw the delayed start patients, those who received placebo initially, have the same target engagement and the same similar reductions in neurofilament, and you could start to see the emergence of and the separation in the clinical data. So really that then lent us to think, well, where are we getting our substantial evidence of effectiveness from? And it was very, very clear that it was on the biomarker, right? So when you see those substantial evidence of those substantial reductions in neurofilament, We then had all of our work cut out for us to then show that in support of an accelerated approval, that those are then reasonably likely to predict clinical benefit. So there's an extensive research group within our company who were looking to really model that so that we could successfully position the NDA for the FDA to begin the review as a potential accelerated approval. But of course, we're doing this whilst we're building the NDA, right? Whilst we're writing all of the documents and all of the summary documents and having a lot of internal discussion. And then we really came to the agency at our pre-NDA meeting, right? If those of you who've been involved in this, it's often a little bit more tactical. And that's where we asked the most strategically important question of all of it, which was, based on what we can share with the agency at this early part, could those tofersen-driven reductions support an accelerated approval? And that's a tough ask for the FDA as well, because they've got a limited insight to this data. And I think having the benefit of the open label extension as well controlled was certainly helpful in that, to have that time course. But then really all the fun was right ahead of us. And because, I really credit Dr. Frelich and Dr. Baracchio and especially the ClinPharm team at FDA who were doing their own assessment on all of this data, right, to understand are the tofersen-driven reductions in neurofilament really reasonably likely to predict clinical benefit? And so we're all building this at the same time, really, and obviously responding to the agency's information requests and under priority review as well, right, which is a tough ask on the agency side as well as ours, whilst also then preparing for the advisory committee meeting. One of the other parts of this program that I think was really important that Steph and I had spoken about internally at length was we knew the advisory committee meeting, we knew we would have an advisory committee meeting, the PCNS committee meeting, at some stage. But we always felt it was probably the most important day for the program up until it became approved, because to really understand the tofersen story and the synopsis that Steph provided, you kind of need to see it all as an NDA. And we really felt that the advisory committee meeting, whilst I prepared for them before in oncology, we kind of dreaded them a little bit because they're kind of scary, right? There's a lot riding on that one big day and it's public. And this one we were really looking forward to because it was a chance to tell the learnings from the study, of how we would design the study differently, right? To be really transparent about the data set we had, how we believed we could show substantial evidence of effectiveness, and to hear from FDA and obviously the committee members themselves. So I think throughout that whole process, there's a lot of learning, a lot of internal debate, a good team that can challenge each other. Steph and I, that was mainly our night shift pretty much. When there was a day shift was operationally getting through the programs, and our night shift was outcome strategy and working through the different data sets. But a remarkable time and a remarkable way to really try to optimize as much of the regulatory toolkit as is available to us.
Joe, you add, or you good? Do you have anything you want to add?
I can add a little bit. I think that, and again, Teresa talked about this yesterday as well in the opening session, but it's exciting to have a biomarker be reasonably likely to predict clinical benefit in patients with ALS, but there are caveats, as we talked about, as to why this was possible for this population. And I think the review, shed light on everything that Stephanie mentioned. This is a targeted therapy for a mutation. We had evidence of target engagement of that, ASO on its target, and then the plausibility that the neurofilament light is a downstream effect of reduction in SOD1. And that, along with the data, which again, when we come back to substantial evidence of effectiveness, we had a robust finding on NFL that was persuasive, and the confirmatory evidence was the reduction in SOD1 showing the target engagement. And that together was enough to kind of get it to the finish line for accelerated approval. I think neurofilament light, as Dr. Baracchio said yesterday, is not ready for prime time. It's not going to solve all of our problems, but it is a really compelling biomarker, and it can tell us a lot about disease. I think the more natural history data that we have on NFL in each disease state would open the door for discussions on when it could be helpful. As was mentioned, the data that has came out in the few years since the tofersen trials began and when it was approved really kind of changed the landscape and our understanding of NFL a lot. And our team, the ClinPharm team, really dug into that literature and how NFL could be used prognostically, and really helped us come to the determination that it was reasonably likely to predict clinical benefit.
And I think this, I would just say, and put a bit of a plug, I think at CPATH, one of the things this did was it put up a bit of a flare because of the conversations that have been brewing in other disease states around NFL. So we're discussing, amongst even our liposomal storage diseases, everything to ALS, is just to look at, a deeper dive of NFL and what we can, and under specific contexts of use, explore. So kudos to that. Allison, I wanted to touch base with you next, and obviously you've been involved in some recent approval processes, and I wondered if you can share some of your experiences and things that you can say that are within the public domain, and see your action with agency or things, the pearls that you might talk about for building your totality of evidence and
Sure. Yeah, thank you. And thanks for putting this panel together. It's great to hear about everyone's experiences and what leads to totality of evidence. So I am able to speak to a recent product approval for a rare disease with significant unmet medical need. Our approval was based on one adequate and well-controlled trial plus confirmatory evidence. And we were able to establish that evidence in a pivotal trial with confirmatory evidence, which included strong mechanistic support, well-established etiology of the disease, the mechanism of action of the product, and also biomarker data. So a lot of that played into the approval. But there are a couple other success factors that I think are worth mentioning. We did receive breakthrough therapy for the product because it was a rare disease and unmet medical need. And that was really helpful because breakthrough therapy allows for increased communication with the agency, and the the expedited programs and pathways at FDA are really important in this context for encouraging rare disease drug development and making sure that treatments make it to patients as efficiently as possible. And when you're dealing with the challenges, which I know we all know very well, of rare disease drug development, the small patient population, heterogeneity sometimes, it's really important that those clear lines of communication are there, and that industry isn't heading off on the wrong path regarding the use of certain clinical trial design or statistical analysis plan or the particular biomarker that you're using. You can have a drug that works, but a bad study design and your trial fails, and nobody wants to be in that situation. So it's really important that people, as you're able to, use these expedited programs and pathways that exist. And we were able to take advantage of that increased communication throughout the development process, and there were instances where we did need additional guidance from the agency, and we were able to get that in a timely manner and address any needs that were brought up, and in a way that we did not have to delay our trial or our timelines. And so that was really important in making sure that product made it to market as efficiently as possible. Another huge piece of this was the patient voice, and the particular disease area that this product is for had an extremely strong patient community. And in this instance, 2 patient groups came together to develop patient perspective data on the disease, the impacts, the symptoms, outcomes of importance to them. And they did it in a very rigorous way, and they submitted it directly to the agency. And the FDA was able to use that information and mentioned it to use it as a additional source of information in their benefit risk analysis. So that was really important, and making sure that you're engaging with the patient community and really partnering with patients throughout the drug development process, not at the end, but early in the process and throughout the process, and making sure you're understanding what outcomes are important to them. And kind of related to that, I mentioned the biomarker data, and FDA has said this time again, and guidance is, and when you have your biomarker data, it's important to be able to link that to what's important to the patients. It's not just a measurement. You need to show how that measurement is clinically meaningful and how it's impacting your patient population, and making sure that that impact actually matters to them. So that's another key piece here. So overall, I think there are, in summary, three things, and that's early communication, frequent communication with the agency, showing how a change in your biomarker impacts outcomes of interest to patients, and making sure that you know what those are when you're developing the drug, and you really cannot understate the importance of partnering with patients and working with the patient community. Patient-focused drug development and rare disease drug development go hand in hand, and I think that's the way that it should be. Making sure that you have that data from the patients. Patient groups are extremely savvy. I know several people in this room have been involved in a lot of those activities, but they can really gather the data in a way that can support regulatory decision-making, and that's a really cool thing to see when it happens. So partnering with the patient community and making sure that they're partners throughout the process.
Dawn, just to add to that, you've got some examples of using endpoints and different special populations. I wonder if you could elaborate on that, given the patient communities.
Yeah, I want to speak a little bit to feel, function and survive, and how you can use the same measure, but your responder definitions can potentially look very different depending on the pediatric population. So I think it's been clear already that we need to have such a clear understanding of the natural history, both naive and on standard of care. We have to understand the disease stage and the phenotypes very clearly related to what each of those phenotypes is going through. And so we're studying MPS diseases at REGENXBIO for gene therapy. And I wanted to share a little bit about how a responder definition, even within a specific disease and a diagnosis, you sometimes need to stratify in order to answer that question. And so we have neuronopathic patients that we obviously want to treat as early as possible because it's a neurodegenerative disease that's progressive. But at the same time, when those children enter into the trial, they have minimal disease symptoms. And so a responder definition for that group of kids may be that they're maintaining their developmental function within a range over time because we've caught them early, right? And then similar children in the trial who have more severe baseline disease presentation could look very different, and we're assuming that we're not going to have the same level of change in both of those groups, and we're going to be looking at very different and maybe smaller changes in the group that has more severe disease presentation at baseline. So if I take those same outcome measures and I look at them in a different pediatric disease and say, how does the responder definition change? So when I was working at Alexion on hypophosphatasia, Strensiq was the drug. And it was the exact opposite there. The most severe kids in that indication were the very young ones. And they had, minimal bony integrity and muscle strength to move. They were ventilatory dependent. And feel function and survive was very different for that group. Those kids, the responder definition was survival. It was very different, and any skills they acquired was a bonus because of this incredibly severe disease presentation they had at baseline. Whereas the flip side, there was a attenuated or late onset version of hypophosphatasia in which we had to have a completely different approach in the outcomes. And in the younger group, it was easy for us to understand, and the regulatory bodies to understand, that survival and not having a control group made perfect sense for that group because it was obvious that they looked different than the natural history. But for these older kids, they came back to us and they said, no, this doesn't apply here. We need more evidence to understand how this group of patients looks different than the natural history. And I think in following that feedback, we were able to, fortunately, we had video recorded all of our assessments on ambulatory function. good backup, right? That was a very good thing for us. And the PI that worked in that trial had been doing longitudinal video collection of ambulatory function on his kids for years. So all of a sudden, we had an opportunity to do central data analysis of these videos and meet this requirement to have some level of comparability between these less severe patients and the patients that were untreated. And so I guess my point here is that the regulatory feedback is incredibly important, but you also have to have such a sound and detailed understanding of the disease stage and the phenotypes, because one responder definition is not going to work across these groups. And it's independent of where you position your endpoints, right? It doesn't matter if you're going for accelerated approval or you're using one of these functional measures as your primary endpoint. You still have to have a high level of evidence and confidence in either one that you're showing meaningful, clinically important change, especially to the caregivers and the families. And so I think that's the lessons learned, is dig into those early stages, really use the FDA guidance and the roadmap to really understand. And people tend to jump to this end of the continuum and say, what outcome can I use? And I think you got to go back to your basics and just understand every nuance of how those kids function in the different groups and how that changes so subtly, whether they're, you know, in the delay stage, the plateau, or the decline stage. And so that's
I'll
thought.
open up to others. I mean, endpoints are commonly a challenge in rare diseases, especially if there's been no regulatory precedence for their acceptance. Does anyone else on the panel want to talk about their positive and benefit experience around specific endpoints? I had to laugh and Michelle were looking at each other when we talked about the roadmap.
Roadmap, yeah. I think everybody at Regenix does not want to hear me talk about the roadmap anymore.
I want to make a plug for a program that FDA has recently started in relation to the endpoints. We now have a new pilot program. It's called Rare Disease Endpoint Advancement Program. And we started it as a pilot following reauthorization of the Prescription Drug User Fee Act last fall. The program is up and running. We put information about it on our website in painstaking details. It explains what information is needed, what aspects of endpoint development would be important, and I invite everybody to visit that website, and if you put this Rare Disease Endpoint Advancement Program, you will see it there. We invite to send us proposals, and we receive a proposal per quarter, initially as a pilot. So that's the plug. Some assistance from the regulators on development of endpoints.
Yeah, just one comment, which is, I think a lot of times, we were just talking about this, I think a lot of times we focus on what are the right endpoints for our trials, and will FDA accept this endpoint? And I was saying this in the context of our AMP-ALS conversations. We've got to take it way back. We need a few steps. What are the measures, fluid biomarkers, clinical outcome measures, of how, you know, that are relevant in the disease, kind of carrying on from what Don was saying, at different stages of disease? And can we refine those measures? Can we identify new measures? And then we can figure out how to use them. Because I would argue that while neurofilament has obviously played a really important role in the approval of tofersen in the context of the surrogacy story, equally important is the role that it played in helping us control for heterogeneity. I mentioned how we got our enrichment strategy all wrong, and you can actually be more inclusive in clinical trials if you have a measure that is truly prognostic. And so I think that when we're talking about endpoints, sometimes we lose our way a little bit when it's really, let's bring the most objective and sensitive measures forward in different contexts of use.
Can I just add one point to hers? I think the next biggest challenge is we want to work towards shortening the time in development process and using accelerated approvals when we have the opportunity. And some of these biomarkers have tremendous opportunity to work across the lifespan, and we can see change across all of our patients, which is great. But then we also have to show that it's reasonably likely to predict clinical benefit. And that's where it gets very tricky based on what I said, because the magnitude of change for that young child that's in a normal range at baseline is going to be completely different than the magnitude of change that is going to be present for the child who's very severe at baseline. And that limits your ability to show this beautiful, strong statistical correlation because it's going to be very different depending on those two groups. So I like the idea of having this one measure that you can use across your population, but then you still have to very much understand what are you going to do with that to show what is reasonably likely to predict clinical benefit and how it looks different by groups.
So we'll switch gears a little bit to Jesse. You've got an exciting gene therapy, SKYSONA, if you want to talk about that experience.
Yeah, so as I mentioned in my introduction, I spent some time at Bluebird Bio. I was there for 5 years, and during that time I worked on a product for the treatment of cerebral adrenoleukodystrophy, which is now known as SKYSONA. I want to be clear that I was not at Bluebird Bio at the time of the submission of the BLA nor the approval. However, I did spend 5 years of my life working on the regulatory strategy leading up to the BLA. So SKYSONA is an LVV-based gene therapy, and it received accelerated approval in September of 2022. So the accelerated approval was based on an intermediate clinical endpoint, not a surrogate endpoint. And since I was there for the 5 years before, I can tell you that we never contemplated accelerated approval for this program. We had agreed with the agency on a clinically meaningful endpoint of major functional disabilities, which was the top 5, I think it turned out being 6 at the time of approval, deficits that these patients may experience. They were all drawn from the neurologic function scale. So our clinical development program and our primary endpoint was against natural history. So the clinical development program included 2 natural history studies. There was one natural history study, which was a retrospective natural history study, and the data that was collected there was not contemporaneous with the conduct of the registrational studies. And these patients were untreated patients. And that group of patients was intended to perform a benchmark against which we would compare the primary efficacy endpoint of major functional disabilities. There was also a second natural history study that was performed. This was a prospective natural history study, and it included patients who received allogeneic stem cell transplant for their CALD, which is the standard of care of treatment for patients who do have a match. And this study was run contemporaneous with the registrational studies, and we used the same endpoints, the same sites, et cetera. In addition to these natural history studies, there were 2 registrational studies, which included in excess of 60 patients with cerebral adrenoleukodystrophy. And per FDA guidance, all patients treated with SKYSONA were followed for a total of 15 years following their initial treatment. So there was also a long-term follow-up study. So in this example, FDA granted accelerated approval when this was not initially envisioned. So during the course of the BLA review, FDA had concerns about the ability to interpret the primary efficacy endpoint. And one of those concerns related to the comparability of the natural history population compared to the treated population. And specifically that the natural history population was more severe in their disease than the treated population. And so as I mentioned, this was data collected prior to the conduct of the registrational study. Treatment methods had changed with time, and we didn't have the same diagnostic methods, patients were not being screened at birth, for example, and we didn't have the same genetic screening methodologies. Given that, FDA requested that subpopulation analyses be performed. Those results bore out the overall conclusion that there was a benefit on major functional disability, but in a much smaller population of patients. In fact, over 80% of the SKYSONA treated patients were excluded from the subpopulation analyses and aren't included in the SKYSONA label. So I think this case study illustrates some of the challenges in rare disease development that many of you guys have touched on today. Sometimes there is the need to leverage historical or prospectively collected natural history study. Sometimes it's not even possible to collect that data. And even if you do collect that data, which Bluebird Bio did, it may not be sufficient in terms of being able to well match patients, just given the size. So, I think there's a lot of power in what CPATH is doing in aggregating data, right? So if it's just a company conducting a few natural history studies, it certainly doesn't have the same numerical power as many companies putting things together, or a single database that has a lot of data. I think in this case, or in all these cases, I think regulatory flexibility and allowing analytical methods to adjust for natural history and treatment group differences for rare diseases could be really helpful, especially when the landscape is constantly changing. And the first accelerated approval for a gene therapy actually just occurred in June this year with Sarepta's DMD program, which is very exciting for us in gene therapy. And we hope that the agency will be more open to the use of a surrogate versus an intermediate endpoint. There's a lot of conversation today too about interfacing with the agency early to discuss these things. And I think there's often hesitance to have a discussion about whether or not something's can be a surrogate endpoint when there's no clinical data to support that the idea that there will be a clinical benefit based on the endpoint. But I do think that gene therapies are a space where it makes a lot of sense because the causal pathway of disease is clearly known. I think that their utilization of biomarkers as surrogate endpoints is definitely a reasonable pathway here. But for each rare disease, it's generally a novel surrogate endpoint, which adds challenges, unlike in oncology where, there are established surrogate endpoints that have been validated. And with time, I'm hopeful that the agency will also start to consider things like non-clinical data as supporting the idea that something could be a surrogate endpoint. But time will tell with that. So they specifically said in the Sarepta basis for approval that non-clinical data did not establish something as a clinical correlate. So anyway, that's my thoughts on Scarsona.
Larissa, do you have any comments on?
Sure. First of all, I would like to make a correction. Apologies for
no problem.
the first approval, accelerated approval of gene therapy happened in 2020.
You're right, it was
And
a
that was,
CAR-T.
exactly,
Apologies.
and that was no problem. And
Yeah.
that was chimeric antigen receptor T cell therapy for treatment of mantle cell lymphoma. But as I was listening to the case studies and stories of different development programs, NDAs and BLAs, I was actually thinking of a proverb and the consequential aspects of that. And the proverb is all happy families look alike, but every unhappy family is unhappy in their own way. And so we're hearing today about happy stories, about successful product development programs. And they all have common features. And I think it is important as a takeaway message today is to understand and recognize all of these common features, because in preparation for future development programs, all of them are important. And I think each speaker actually talked about various aspects of their product development programs that are in fact applicable and would be counted in that list of common features for a successful development program. And that includes very good understanding of the natural history of the disease, genotypes, phenotypes, heterogeneity of the disease, the different types of progression in different subtypes of the condition that you're looking at. Very good understanding of what it is that one is intending to treat, and a great understanding of how a potential biomarker or a potential surrogate endpoint can actually fit in that natural history of the disease and what it can measure and how it can measure it. And one part that I haven't heard today and that's still in surrogate endpoint biomarker development piece of the success, is a really good validated assay for how the biomarker is measured. Because we see oftentimes biomarkers that make sense for product development, make sense for the natural history of the disease, even may be acceptable in some clinical practice measurements. But if you don't have a validated assay of what it is that is being measured, particularly if that biomarker is then eventually going to be used as an endpoint, then there may be an issue there. So measuring biomarkers in the way where you can have a reliable measure, and best if it's validated. Engagement of patient partners, that's also one of the crucial features of a successful development program. Understanding what it is that needs to be put into the package for substantial evidence of effectiveness. Obviously, for rare diseases, the agency has said many times that instead of 2 adequate and well-controlled trials, we're looking at 1, recognizing the rarity of the condition, looking at supportive evidence, looking at how the study was conducted, looking at study design, video assessments, very well thought through instruments of how different, be them clinical endpoints or patient-reported outcomes, how they're going to be assessed, right? So good instruments and study conduct where data integrity is preserved. So that's another feature which is important. And specifically related to gene therapies, with cell therapies too, biological products, many of them, there is a term that someone coined, and that's a living drug, right? In many cases for gene therapies, it's a single administration. For cell therapies, it may be repeat administration, but even with the single administration, we do expect longer effects of that treatment to be there. So a longer time of observation may often be needed. And sometimes you can't really make it, be it with surrogate endpoint or with an intermediate clinical endpoint, by the time when the product is approved and on the market, you would still want to see what are the long-term effects of that therapy. People talk a lot about curative therapies. When do we establish cure? We have to really look at the absence of the relapse, at change in disease progression to the degree where there is no progression anymore. And the principle of faster, cheaper, better works best with semiconductors. But when we're working with biological systems, you really want to make sure that the effects of the therapy, the intended and the anticipated effects of the therapy are there. We had a meeting with stakeholders on April 27 of this year, and we talked about various methods for how one would evaluate both efficacy and safety in the post-approval stage, particularly when we have many products that are in development and people do not want to wait until the product is available to them. So we're looking at shorter times for development programs. Again, even if it's not with a surrogate endpoint, if it is with an intermediate endpoint, you still need to see the longer term efficacy. And we've received a lot of good feedback from those who participated in the workshop. We're planning on developing a guidance, on the topic. But one interesting part that I, took from that, we have certain time frames for how different product categories would need to be followed over time for, say, lentiviral vectors, 15 years, right, you mentioned. For some other vector-based products, it could be shorter period. But what we've heard from stakeholders is that they are concerned not just with understanding of the efficacy and safety, but also was life events with life milestones. For example, effect on fertility. If somebody receives their gene therapy very early in life, in the first few years of life, even the 15 years that are there are not going to cover the period when they are of a childbearing age. And so again, with development of technologies, we all have to think about how we, follow people and better understand the effects of various long-term products that we give to people and how they affect their life. Yeah, I guess that's
good. Jesse, did you want to comment about post-marketing requirements for gene therapies in relationship to the discussions that you've had?
Yeah. Sure. Yeah, so for Skysona, which I just spoke about, there are 2 studies that need to be performed for the accelerated approval. And one of those studies is to enroll 24 newly treated patients who have more advanced disease, which may be a very big challenge considering that there is newborn screening efforts, there's genetic screening. The likelihood that a patient today will be treated later in the course of disease is much lower than it was previously. So I think, sometimes there are challenges associated with the requirements that are put in place for sponsors following approval. In addition to that study, there is a post-marketing requirement as well to conduct a prospective observational study in 120 patients, which needs to be completed in 2047 to assess long-term safety risk as well as the risk of secondary malignancy, which did appear with Skysona. That as well is, I think, a big challenge and ask, considering it's anticipated that approximately 10 patients per year will be treated with Skysona. So, that's 12 years of patients for enrollments. And I totally agree with everything you're saying, by the way. I'm not, but I do think that we need to, because sometimes I think accelerated approvals occur later in development versus, discussing upfront and early, like that you're headed towards an accelerated approval. I think sometimes there isn't time for conversation about what would be a meaningful study and time to allow the sponsor to sort of like undertake and think through that more. But at any rate. Sorry.
Yeah, I guess I just wanted to maybe discuss a couple of items here. One is dealing with heterogeneity of the disease, right? So that's another kind of, continuing on the same topic, common feature of successful development. So you have a very heterogeneous disease. There is an effect in one subpopulation and maybe trend in another subpopulation. You really would like to see what that effect looks in the subpopulation for which you don't have sufficient data. And so here we have this ability to monitor the other subsets of people, the subgroup with pyramidal tract, right, disease, and the other subpopulation. So there are ways of how one could look at one heterogeneous disease, maybe see initial efficacy demonstrated in one patient subgroup and follow other subgroups over time to see what happens with the treatment, in those people. So that's one. And for the safety requirement for the post-marketing study, I'm sure you know well, Skysona did have a signal. It did have a signal with myelodysplasia observed, and so we owe answers to people who are being treated or who are potentially considering treatment with novel therapies. And when we don't have enough data about safety signals that are seen in pre-marketing, we try to follow those patients and try to follow those who are treated with the product to see what is the actual incidence and frequency of potentially very devastating side effects, because obviously with gene therapies, with lentiviral vectors, we are worried about secondary malignancies.
And, if it takes a certain number of people or a certain period of time to understand if the risk of second cancer is there, I think many people have already answered this question as, yes, we do need to have follow-up.
No, it's just to say, around the whole accelerated approval strategy, before I was working on tofersen, every oncology asset I worked on, accelerated approval was our only strategy, primarily, right? Because you're targeting a late line indication, but there's just so many differences that I think are important to point out before we get to the similarities, is because virtually all oncology accelerated approvals are based on some form of response rate, right? Durable response rates in some form, and that is exactly what you're looking for in the same data as for your go, no-go decisions or your expansion of phase 1 to have something that's potentially registrational. So when you're going into your first of human study, you're hoping for some partial responses, right, in the heavily pretreated patients, or if you get a complete response, then you start to expedite further. So it becomes part of your primary strategy. I think some of the complexity that we're talking about here when it comes to a biomarker as a surrogate endpoint that's reasonably likely to predict clinical benefit. Often we are having the same discussions with tofersen as we were beginning to determine what was substantial evidence of effectiveness was a biomarker that's never been used for regulatory decision making before, right? So whilst there's some clear differences there in the data and the evidence you need to build throughout development, ultimately I think it comes down to the same criteria when you try to understand what is reasonably likely to predict clinical benefit. You have your biological plausibility and your empirical evidence. I completely agree with what you were saying about the need for robust assay development, particularly as the effect on a biomarker becomes less, then of course you need an even more kind of robust assay. So I think it's fascinating. I think it's the most challenging is for when you're going first. I worked on assets in small cell lung cancer where we had overall response rates that is used commonly, but in that particular indication, when you have a tumor that's doubling in size every few months, it's not reasonably likely to predict clinical benefit. So there's so many learnings, I think, in the successes and also the products that don't make it. But it's complex, I think, and when you're talking about a biomarker to be used in regulatory decision making, you often don't know until you get there, until you know what your empirical evidence is, and you're still working through the biological plausibility. And that was part of our advisory committee meeting strategy, was to try to kind of weave that tapestry together to say what is reasonably likely and how are we demonstrating it.
Any other comments?
I'll just add, that was a great discussion, and I think, to quote Michelle Campbell, but engage early and often. And, when we're talking about endpoints, whether it's biomarkers and surrogate endpoints or choosing the right outcome measure for a rare heterogeneous disease, the earlier we start that conversation, the better we can kind of help you have a successful study. I think when we talk about assay validation for biomarkers and surrogate endpoints, the earlier you plan to use a biomarker or to rely on an intermediate clinical endpoint for a trial, the better it is, because you can plan for that accelerated approval, and then the post-marketing requirements and the things are easier to successfully complete. I think we heard 2 stories today of kind of last-minute accelerated approvals, but they're most successful when you know that early going into it. And then sometimes that study to confirm clinical benefit can be almost done, almost completed by the time you reach accelerated approval, and you don't have to worry as much about how do you complete that trial in the face of an approved product and, dropouts and enrollment and recruitment. So I think if you can engage early, the better you'll be. And I just wanted to clarify one point that comes up a lot in just terminology is confirmatory evidence versus confirming clinical benefit after an accelerated approval. And they're 2 very different topics.
I just want to take us back to the patient piece for a minute. We saw some great examples of how you incorporate patient/caregiver input into your drug development pathway. And I think industry as a whole tends to be really good at capturing the patient diagnostic journey, the patient journey. I think that's important, but I think that's only a tiny piece of what we should be doing with caregivers. Meaning that I think we're totally underestimating what they can do, how involved they can be, and we should be asking them the really hard questions. They know the diseases better than any of us, and if you come to them and say, what is the relevance of these items? How do these items, how meaningful are they to you in your world with your child? They will give you incredible examples of the item relevance and how that impacts them in their daily life with their children. And that's part of your outcomes. That's part of your narrative and your story. And the other example I think that should be highlighted is PP-RND and their patient preference work. And so in gene therapy, the work that they did on a large sample of caregivers related to the level of risk that they're comfortable in, how that differs by phenotype and disease progression, it was so informative and so important, and that's a huge example of how everybody in the DMD space can benefit from understanding how caregivers make decisions related to enrollment in a study. And so I'd like to see us all kind of shift away from going to vendors and seeing these beautiful PowerPoints on the diagnostic journey. That's great, but that's not the real questions that we should be asking of these caregivers. We should be digging in and really using them as a resource that's powerful and meaningful and can really change the direction of our development pathway.
And to add to that, I don't think it should really stop there. You know, when you're looking at post-marketing requirements and follow-up studies, you know, you still need to engage with the patient population to reduce patient burden and help make sure that you're actually you're designing that follow-up in a way that you can get that information from patients and really help retention. So it doesn't just stop with approval.
Right. Sorry, just back to the SKYSONA for a second. I didn't mean to uncover two contentious sides of the sponsor FDA claim. I do, just in terms of patients, want to say I do think that FDA exercised an incredible amount of regulatory flexibility in finding a path forward for SKYSONA so that patients and caregivers can make an informed decision about whether or not this is the right product for their patients, but it is available. And I do think FDA was incredibly creative, and it certainly is not the perfectly executed program. There absolutely was a MDS that appeared and a risk of malignancy, and gene therapy should not be taken lightly, right? It's a one-time treatment. You can't get something again, So at any rate, I think FDA did do a really amazing job in terms of the regulatory flexibility that they exercised there.
Yes, I'm jumping backwards, but this has been on my mind quite a bit lately, and it's probably an obvious statement, but I think as we're thinking about new endpoints, and using ALS as an example, but this isn't unique to ALS, so much of it comes down to what you're anticipating with the therapeutic landscape. And as we look at ALS, when you look at not Biogen's work, but the whole community's work of therapies that are in development, and as we understand the disease better, as we're developing more targeted therapies, we've always sort of operated in a world where what we were hoping for was slowing of decline. But if we identify and develop therapies that can actually lead to recovery of previously lost function and strength, the existing gold standard endpoints are not going to be sensitive to assessing what, I'll coin improvement across those measures. And there are some really interesting examples. We had physical therapists that were looking at tofersen-treated individuals and seeing things that we were not seeing with the endpoints we were measuring. And all of a sudden it made me think, these are endpoints like the 10-meter walk test, the TUG, the Berg balance test that are being used in many of the diseases that we're talking about today that we had never considered in ALS. And maybe that is something that we could learn from each other and really try to talk more cross-TA when we're considering new endpoints in the future.
I guess I'll open up for any questions. We're using up all our time, so I want to make sure that if people want to give a chance to discuss some things, please up to the mic.
My question is about for rare disease. So we might have some clinical trials that are global in EU and in the US. So in having this dialogue with the FDA, so should we have a dialogue with the FDA separated from the EMA? Or is there a kind of coordination if one, for example, refuses to have a liquid biomarker as endpoints and the other authority is okay with it. So how does it work? Is there a coordination? Should we talk to both separated? So that's my question.
So I'll try to address this. Obviously, because we're in different regions, each country or different countries, EU includes a number of countries, they have their own laws, and regulations are extensions of laws, so the regulations are different, obviously. In many cases, particularly for rare disease, we try to collaborate and converge on the requirements for product development, but each country is still under their own regulatory framework. However, because many studies are multinational studies, they're multi-regional studies, oftentimes what we see is that the same one study can be sent in to both FDA and EMA, and for example, right, maybe to other regulatory authorities too, but I'm just talking about FDA and EMA. Sponsors typically have separate meetings with FDA and EMA under processes for each of the regulatory agencies. We do have a process that's called parallel scientific advice with EMA when we can provide parallel advice, yet it is still not the same advice. It could be converging, but wouldn't necessarily be the same. In some cases, it could be very similar, if not the same, but it's not really the same advice. It's just to make sure that the requirements for each region have been met and not at the expense of, doing something very large or not feasible. So we try to work together with other regulators. We also have various bilateral meetings where without sponsors we can discuss development programs for different products and just exchange opinions on what could be done or what has been done and how we assess different development programs. We have clusters in different areas in patient advocacy and rare diseases in various therapeutic areas. So we do collaborate to an extent, but again, every region is under their own regulations.
Hi, my name is Yin YIN from Belgium. I'll say hello to you guys after the meeting. I have a question about for the FDA, for the questions you post for the advisory meeting. I do not watch all the advisory meetings, but I did watch the Tofferson because it's about, and I think the first question was about the accelerate approval, and it was very clear, and you got unanimous votes for that. But there was a second question, and the question was asking the advisors whether the evidence was significant. And then a lot of advisors kind of were stuck by this significance. And then the question changed from significance to convincing. So they still had the same trouble with the adjective. So I'm a statistician. I think I try really hard when I work with teams, try to avoid using adjectives, use numbers. And because, because when people say, oh, it takes too long, how long? Years or days? Or when they say expensive, how much money you're talking about? Sample size, or too many people or too few people? How many are we talking about? People have different assumptions. So I think the question was kind of using adjective, just really confused a lot of people. Even for English major, probably they have trouble to understand what the difference between significance and non-significance. And I think FDA Clean Farm presentation did a good job, and they have this good story and incorporate all the numbers to have this package. And then I look at it, two people voted for yes. And their reason was if you even approved by this accelerated approval, the patients can still not afford the drug because if not the full approval, the insurance is not going to cover it. So it's really kind of, so those two advisors really kind of stand from the patients kind of in their shoes and to think about it. Other advisors were thinking about what convincing means. So I think that the question should be maybe like, are you going to prescribe this drug for your patients? I think that probably, that's their job. It's not to interpret significance or, adjectives.
I appreciate the comments, and I think we learned a lot from that advisory committee as well. I think there was the attempt, the application and the discussion was about accelerated approval, and that's how the NDA was submitted and reviewed. And there was a thought that, did anyone on the advisory committee feel that the data was substantially convincing that it should get full approval? And that was maybe, may or may not be poorly phrased, but it was an attempt to see if the data was there according to the committee. And so that was how we phrased that question.
Tim?
Yeah, thanks, Colin. Tim Franzen from CPAP and Faegre Drinker. Thank you all for some really instructive comments about best practices. And that's always important in learning. Something that we may benefit from as well as the converse. So experiences you would have had that show the need for regulatory inflexibility that are unreasonable, and the opportunities to learn from those. So would the panelists care to comment on that?
We have to bear in mind who we have in the room and on our panel as well, I think. But it's a good question. I think that this came up a little bit when we were discussing the accelerated approval reform, right? A little bit about, are we taking enough risk with the regulatory pathways, right? And then you can inherit from there your question, really. I think we did a lot of work as a company, and as an industry group, to assess successes of the accelerated approval pathway. So programs that were approved as an accelerated approval, then went on to confirm benefit. Then we looked at products which did not confirm benefit, and another regulatory decision was either made to withdraw or in some circumstances leave them on there. And I think looking at it from that lens, you come up with, if you will, about a failure rate of about 8 to 10%. So you can see for all accelerated approvals from 1992 onwards, right, the failure rate is about 8 to 10%, i.e. programs that gained an accelerated approval then didn't confirm benefit. To me, that's quite a good metric to look at. I mean, if that number was 2%, right, then I would say maybe we're not taking enough risk with the pathway. Then that would suggest to me that maybe there's too much inflexibility, right, around what is reasonably likely to predict clinical benefit. Conversely, if 40% of them did not go on to confirm benefit, then maybe we're being a bit too liberal with how we're defining reasonably likely. So just from that metric, to me, 8, 10, 12% feels about right, holistically across all development programs. So I think it's case by case. That's accelerated approvals are typically on the more aggressive end of regulatory strategies, and I think that's a good way to look at success and failures, to look at that kind of generational impact of that piece of legislation that enables that. I often hear folks asking for more consistency in regulatory decision making. I hear that mainly from industry. It's not a point of view I subscribe to. For all the examples we just discussed today, there's so much nuance in there, right? There's so much that the FDA has to review that is completely new data, completely new context of use, often under a priority review, because these are the more serious, life-threatening conditions of highest unmet medical need. So I think it's a difficult balance. So I think you have to take a step back and look overall. Metrics will tell you one thing, right? But in terms of enabling FDA to make decisions, right, that's on industry as well, right? To be, as we were discussing, how is one defining substantial evidence of effectiveness? And if we had not been successful with the tofersen application, I would look at that as a failure on our part, that we haven't enabled the agencies to make the decisions they're doing. So it's a difficult question to answer. I'd look at accelerated approval metrics, but Maybe others have other thoughts.
If you wanted to say
One
it,
comment,
I'll
yeah, and I'm not just saying this because Dr. Freilich is sitting next to me, but I always say to the team, agency knows more than we know, right, about more products. And I love the example that when we were designing the phase 3, we were talking with the division about the sample size and requiring a two-sided alpha of 0.05, and I said, I don't think we can enroll 100 people. And I remember Dr. Dunn saying, you need to try, and then we can have a conversation. And in hindsight, had we not tried, had we not enrolled 100 people, I don't know that we would have been able to interpret the data set. So it's one of those good examples. I wasn't excited about the feedback at the time, but I think, again, trusting sort of the process and keeping all of us, keeping each other in check, I think is really important.
Emily, did you want to say something?
Yeah, I was just going to say, we try to be flexible when we can, but we still need to meet that substantial evidence of effectiveness. And I think we don't pull it out of the air. So, when you talk about inflexibility, we are going to stick to the data that's needed to show that a drug is both safe and effective.
I think we need to be cautious in interpreting successes and failures in previous filings in terms of how they apply to us, because I think it's a new world. Rare disease is different now. Like, I've been doing rare for 20 years. The knowledge people have, the approach the FDA speaks to is very different. And I also think we're doing gene therapy, and that's a brand new world. And so I think we have to believe that we're all learning together, and it's a work in progress, and we just need to take it one step at a time and do your homework, right? Like, that's my takeaway. Like, there's no golden ticket or easy pass that you're going to get. There's tons of guidance documents out there that will guide you. And yes, we can't meet all of the boxes in those documents, but that's an okay place to be. That's an okay place to say, you're asking for this, and this is why this doesn't work in my disease or in my subgroup or in this model. And this is what will work. And so I think learning together and using those guidances, and our job is really hard in Rare. This isn't a walk in the park, but so is theirs. And I think we just need to do this journey together and take it one step at a time.
No, so I'll give you the last comment.
Yes, I just wanted to say in relation to the accelerated approvals metrics, the accelerated approval pathway has been there in the regs since 1992. We haven't really used it as much as we're using it now, and I think everybody recognizes that with the change in the landscape of product development and with more rare disease product programs entering the regulatory field, we're seeing an increasing number of accelerated approvals. And I think one important piece to recognize is that once the accelerated approval happened, the game is not over, right? We have to see the verified and described clinical benefit. And one part of some inflexibility that may have been observed in the past was that when there is no demonstrated and verified clinical benefit, it sometimes is difficult to withdraw the product from the market. And so I think, moving forward, as we grant more accelerated approvals, there is a need for general recognition of all stakeholders, of industry and patients and policymakers, that once something isn't shown or the substantial evidence of effectiveness based on clinical outcomes is not there, then the next step is not go into a prolonged process with FDA in discussing, how we can remain on the market. The next step is to, withdraw and maybe repurpose or redevelop or do other things. So I guess, we will see how this issue would unfold, again, because we're granting accelerated approvals. But the lifetime of the product is important to consider here when we're discussing these FAST programs.
Okay. I want to thank the panelists for all their time and lending their expertise to this group, and I'll pass it on to them.